Greetings to everyone watching this video. In this video, we will be discussing about the process of gametogenesis for males, that is spermatogenesis, and in females, which is oogenesis. But before proceeding further, let us know more about us, Revise Me. So about Revise Me, we offer a plethora of quizzes, multiple play modes with a leaderboard, and from this month we have started a mega quiz session wherein you can attend all the quizzes for free and then check your capabilities for the entrance exams. You can stay tuned to our Instagram page to know more. Now, let us move on to the main content. So we talk about gametogenesis. What exactly is gametogenesis? So gametogenesis, what exactly is gametogenesis? अब दो genders होते हैं और दो gametes होती हैं तो gametogenesis भी दो types का होगा. जो male में gametogenesis होता है उसमें होता है spermatogenesis क्योंकि हमसे spermatogenesis से क्या बनेंगे sperms बनेंगे और जो females में होता है oogenesis और oogenesis से क्या बनता है ओवम या फिर हम इसको egg कहते हैं. अब हम सबसे पहले in the upcoming slides we will be learning about spermatogenesis first and then followed by oogenesis. So let us know about spermatogenesis now. So spermatogenesis. So ये spermatogenesis होती कहाँ पे है? It occurs inside the primary male sex organ which is the testis. Now इसके लिए firstly we need to know what exactly spermatogonia are. So spermatogonia, they are the immature Spermatogonia are the immature male germ cells. So the spermatogonia are the immature male germ cells. Now they are present. ये होते कहाँ पे? We know they are in the testis, but where exactly in the testis? They are lined by the inside wall of the seminiferous tubules. They are lined in the inside walls of the seminiferous tubules. So ये spermatogonia है. Now these we know that these spermatogonia will finally proceed to form sperms, but there are various number of steps involved in the middle. So let us talk about those steps in the upcoming slide. So जो हमारा spermatogonia होता है, it is a diploid structure. So it is a 2n structure. It has 46 chromosomes. So ये जो हमारा spermatogonia है, it is diploid and 2n. अब क्या होता है एज टाइम गोज ऑन ये जो स्पर्मेटोगोनिया है इट डिवाइड्स बाय माइटोसिस टू इंक्रीज इट्स नंबर क्योंकि अल्टीमेटली हमें पता है कि स्पर्म्स हैप्लॉइड है. अब हैप्लॉइड करने के लिए वी नीड टू डिवाइड द कंटेंट. अब डिवाइड करने के लिए मतलब हमें बांटना पड़ेगा और आप किसी चीज को तभी बांटोगे जब अपने आपके पास वो चीज बहुत ज्यादा होगी. सो इन ऑर्डर टू इंक्रीज इट्स अबंडेंस ये जो स्पर्मेटोगोनिया होते हैं they divide by mitosis to increase their number. So this mitosis, the first mitosis which occurs, this is the process of mitosis. It is to increase their number. It increases their number. अब जब इनका number बढ़ जाता है, it is nothing but it is given a special name which are known as primary spermatocytes. Now these mitotically divided spermatogonia are known as primary spermatocytes. इनकी भी प्लॉइडी सेम होती है क्योंकि ये माइटोसिस से डिवाइड हुए हैं तो ये भी 2n होते हैं और इनमें भी 46 क्रोमोसोम्स होते हैं. So now अब we have many primary spermatocytes. So now it's time to divide them. तो अब थोड़े टाइम के बाद what happens is इनमें से एक का होता है division. Now they divide and they divide what? How do they divide? They divide meiotically. So the पहला meiotic division, the first meiotic division, it takes place in the primary spermatocyte stage और ये जो primary spermatocytes हैं they meiotically divide themselves to form secondary spermatocytes. So now ये जो secondary spermatocytes बने these are haploid क्योंकि यहाँ पे क्या हुई meiosis हुई. So these are haploid cells which have 23 chromosomes in them. So now what happens after secondary spermatocytes? Let us know. So ये जो हमारे पास सेकेंडरी स्पर्मेटोसाइट्स बने ये जो दो हमारे पास सेकेंडरी स्पर्मेटोसाइट्स बने थे, these are what? These are secondary spermatocytes. So what happens is, they are secondary spermatocytes, they are already haploid and they have 23 chromosomes in them. So now they undergo another series of meiotic divisions, that is, they again divide. They again divide and they form four equal haploid structures. They form four equal haploid structures known as spermatids. Please note, they are not sperms yet. These are just intermediate steps which will subsequently lead to the ultimate formation of the male sex sperm. So ये जो चीज है, these are spermatids. अब ये भी meiosis से बने हैं. यहाँ भी क्या चीज हुई है, यहाँ भी हमारी meiosis हुई है, and this is the second meiosis which is occurring in the process of spermatogenesis. तो ये हमारी अभी meiosis हुई है. So now these are spermatids and these spermatids are again haploid and they have 23 chromosomes in them. ये haploid होते हैं. यहाँ पे सेकेंड मियोसिस होने के बाद दे आर हैप्लॉइड एंड दे आर कॉल्ड स्पर्मैटिड्स. नाउ व्हाट हैपेंस इज, ये जो स्पर्मैटिड्स होते हैं, ये जो हमारे पास स्पर्मैटिड्स होते हैं, दे मैच्योर. These spermatids, they undergo the process of maturation to form spermatozoa. Now what is spermatozoa? Spermatozoa is nothing but another name for a sperm. So they mature into the sperm by a process, and that process in which they mature into a sperm, this process is named as spermiogenesis. So इस process का नाम होता है spermiogenesis. Now what happens after spermiogenesis? Let us know. तो अब जो हमारे पास sperms बने, they are still embedded in the seminiferous tubules, right? They are not yet released from the seminiferous tubules. So अब ये अभी भी seminiferous tubules में हैं. So in order to release them from the seminiferous tubules, this process of release is given a special name and it is known as spermiation. So spermiation is the final process by which the sperms are released and it ultimately leads to the formation of a haploid sperm. So let us quickly revise the process of spermatogenesis in quick detail. What really happens is, सबसे पहले ये हमारे पास spermatogonia होता है diploid. It mitotically divides to form more primary spermatocytes. These primary spermatocytes undergo meiosis to form secondary spermatocytes, right? And these secondary spermatocytes undergo meiosis to form spermatids. They form spermatids. So what are these? This is the spermatogonia. This is the spermatogonia. This is the primary spermatocyte. This is the secondary spermatocyte. The secondary spermatocyte. And what are these? These are spermatids. These are spermatids. Now spermatids are released from the seminiferous tubules by the process of spermiation after they are converted into sperms by the process known as spermiogenesis. यहाँ पे आप देख सकते हैं डायग्राम में, these are the spermatogonia. यहाँ पे ये है, ये सर्टोली सेल है जो सेमिनिफेरस ट्यूब्यूल के अंदर होता है. आप देखिए ये है प्राइमरी स्पर्मेटोसाइट, ये सेकेंडरी स्पर्मेटोसाइट. अभी आप देखो ये जो स्पर्मेटिड है, इट इज स्टिल अटैच्ड टू द सेमिनिफेरस ट्यूब्यूल का जो सर्टोली सेल है, अभी ये स्टिल अटैच्ड है, और क्योंकि ये अटैच्ड है इसलिए हम इसको स्पर्मेटिड कहते हैं. नाउ व्हेन दे आर फाइनली रिलीज्ड, सो यहां से यहाँ के रिलीज के प्रोसेस को ही हम स्पर्मिएशन कहते हैं. सो व्हेन इट इज फाइनली रिलीज्ड, it is known as an independent spermatozoa. So now in the upcoming slides, let us talk about oogenesis. So अब जो हमारा टर्शियरी फॉलिकल होता है, जिसको हमने बोला था कि इसके अंदर एंट्रम होता है, द फ्लूइड फिल्ड कैविटी, द tertiary follicle transforms into something known as the Graafian follicle. So this tertiary follicle transforms into something known as the Graafian follicle. अब इस Graafian follicle के अंदर it has the secondary oocyte or what we call as the ovum. अब ये जो secondary oocyte होता है, it forms a layer called zona pellucida around it. You please note the names, these are very important. It forms a layer known as zona pellucida around it. अब क्या होता है, in the menstrual cycle, in the menstrual cycle at around 14th day, mid of the cycle के आसपास in females, LH surge occurs. Now what is the LH surge? It is the surge of the luteinizing hormone which ruptures the Graafian follicle. It ruptures the Graafian follicle और इस Graafian follicle के rupture से होता है release of the secondary oocyte. Release of the secondary oocyte in what we call ovum as the common terms, and this process, this rupturing and release of the oocyte, secondary oocyte, this process is known as ovulation. This process is known as ovulation. अब जैसे ही वो गर्ल बेबी बॉर्न हो जाता है उस वक्त पे ये जो ऊगोनिया होते हैं, these oogonia, they are arrested at prophase 1. They are arrested at prophase 1 of meiosis 1. Prophase 1 of meiosis 1 पे ये जो oogonia होते हैं, they are arrested for further development. Arrested for development. So they are arrested at prophase 1 of meiosis 1 and they are arrested. अब जब ये arrest हो जाते हैं तो इस stage पे they are called as primary oocytes. They are called primary oocytes. अब क्या होता है इन primary oocytes पे they are surrounded by a layer of granulosa cells. They are surrounded by a layer of granulosa cells and they are known as primary follicles. Primary follicles. Now more layer of granulosa cells are added. Again a layer of granulosa cells are added. Granulosa cells, and they are known as secondary follicles. Secondary follicles. Now you need to know this that हमारे पास oogonia birth पे कितने थे 2 मिलियन oogonia थे, लेकिन जो हमारे पास secondary follicles बचते हैं ये सिर्फ 60,000 to 80,000 बच जाते हैं क्योंकि from birth to puberty a lot of follicular atresia happens. Now what is this follicular atresia? Follicular atresia, that is many of the follicles get destroyed. These follicles get destroyed from birth to puberty. Now what happens? What is the fate of these secondary follicles? Let us know. अब हमने पढ़ा कि हमारे पास ये secondary follicles थे, these secondary follicles, these were the secondary follicles, और हमने पढ़ा ये बस 60,000 to 80,000 बचते हैं. अब ये जो secondary follicles होते हैं ये ज्यादा layers अपने आप पे ऐड करके secondary follicles बनते हैं. अब ये secondary follicles पे एक और layer ऐड होती है granulosa cells की. Granulosa cells की एक और layer ऐड होती है and they are transformed into a tertiary follicle. Now you need to know a very important characteristic of the tertiary follicle, that it is characterized by a fluid-filled cavity called antrum. इस cavity को antrum कहते हैं which is clearly visible in the tertiary follicle. अब इस stage पे during the conversion of tertiary follicle, this tertiary follicle is divided into an inner theca interna, inner theca interna and an outer theca externa. Externa, why? Because we are talking about the outer one, and interna, why? Because we are talking about the inner ones. What are these? These are basically compartments in the tertiary follicle जिसमें वो divide होता है. अब क्या होता है कि यहाँ पे इसी stage पे when the tertiary follicle is formed, इस stage पे हमारी हो जाती है first meiotic division is completed. The first meiotic division is completed. अब जो हमारी first meiotic division complete होती है उसके क्या products बनते हैं? उसके जो products बनते हैं that is a secondary oocyte and a small tiny polar body, first polar body, जो हमारे वो बनते हैं इसके प्रोडक्ट्स. ये सेकेंडरी oocyte को ही is what we call as an ovum, और ये जो टाइनी पोलर बॉडी है इट इज ऑल्सो फॉर्म्ड अलोंग विद द ओवम ऑन द फर्स्ट मियोटिक डिविजन. सो अब जो हमारा टर्शियरी फॉलिकल होता है जिसको हमने बोला था कि इसके अंदर एंट्रम होता है, द फ्लूइड फिल्ड कैविटी, द टर्शियरी फॉलिकल transforms into something known as the Graafian follicle. So this tertiary follicle transforms into something known as the Graafian follicle. अब इस Graafian follicle के अंदर it has the secondary oocyte or what we call as the ovum. अब ये जो secondary oocyte होता है, it forms a layer called zona pellucida around it. You please note the names, these are very important. It forms a layer known as zona pellucida around it. अब क्या होता है, in the menstrual cycle, at around 14th day, mid of the cycle के आसपास, in females, LH surge occurs. Now what is the LH surge? It is the surge of the luteinizing hormone which ruptures the Graafian follicle. It ruptures the Graafian follicle और इस Graafian follicle के rupture से होता है release of the secondary oocyte. In what we call ovum as the common terms, and this process, this rupturing and release of the secondary oocyte, this process is known as ovulation. Now this is your NCERT diagram giving the detailed comparison of spermatogenesis as oogenesis. To my left is spermatogenesis, the process of spermatogenesis, and to my right is the process of oogenesis. We can quickly see what happens in spermatogenesis at puberty. It occurs at puberty, whereas the oogenesis begins at the embryonic developmental stage. You can see that the spermatogonia undergo mitosis to form primary spermatocytes. Now the primary spermatocytes undergo the first meiotic division to form the secondary spermatocytes, which are haploid. The secondary spermatocytes undergo the second meiotic division to form 4 equal haploid spermatids. Again, they are haploid. And the spermatocytes differentiate, mature, and release to ultimately form the sperms. Now, whereas in oogenesis, what happens is, in oogenesis, the oogonia is there. The oogonia mitotically differentiate into the primary oocyte. Now, during this process, follicular atresia happens. I've told you many times. This process, follicular atresia happens. Now, the first meiotic division is completed prior to ovulation. So now what happens is at birth, there are no more oogonia added, right? So at birth, childhood, and puberty. So at puberty, before ovulation, the first meiotic division happens, which results in the formation of a first polar body and a secondary oocyte. Now, in the adult reproductive life, when we talk about pregnancy and fertilization, the second meiotic division happens before fertilization, which results in the formation of the ovum and a secondary polar body. I really hope that you understood the process of spermatogenesis and oogenesis in detail. I would like you to go and attempt the quiz pertaining to this topic on our app to test your learning of the concept. Thank you and have a nice day.